Fantastic. Thank you, everybody, for joining us this afternoon. I can see those student numbers climbing very healthily on a Wednesday. I almost got the day of the week wrong there, on a Wednesday afternoon. So thank you for joining so punctually. Whilst we just wait for everybody to join us on Zoom, I wanted to do a quick introduction. So my name's Lydia, I'm part of Team Unifrog, and I will be hiding in the background today, making sure that everything is working tech-wise. But before I disappear, I just wanted to let you know about how you can engage in today's session and ask questions as we go through. So you'll see on your toolbar on Zoom, there is the option for Q&A. We do encourage you to ask questions throughout today's session. We will get back to as many of you as we can, but we also have a nice 10 minutes at the end of today to have a Q&A session and answer lots of the questions around biological sciences, careers, and studying at the University of Southampton too. But I know it's not me that you come here to hear from today. So without further ado, I will hand over to our wonderful academics from the University of Southampton to take us through this afternoon's taster webinar. Over to you.
Thank you very much. My name's Amrit Muda. I'm a professor of neurosciences, and I'm going to do the first part of this webinar to tell you a little bit about where a degree in biological sciences can take you. Next slide, please. Okay, so you must be asking yourself, why should we consider biological sciences, and what does biological sciences entail? Why should anybody want to study biological sciences? Well, I hope by the end of this webinar, you'll agree with me that it's very, very interesting. It's challenging, it's exciting, but more importantly, it's rewarding. What you study, the things you study about, can change life. It can impact our society today and tomorrow and globally. It can do that. And it leads to many, many career opportunities that will help our society, not only our human race, our environment, what people will benefit from today and tomorrow and in the future. And we'll show you examples of that through two different programs in biological sciences that showcase what I'm telling you. You will see real examples of how it's interesting, challenging, exciting, and rewarding. Next slide, please, Neil. But first of all, if you're thinking about a degree in biological sciences, you might wonder what actually is biological sciences. And some universities offer biological sciences as a degree program, others offer other programs within the School of Biological Sciences. So we are the School of Biological Sciences, and within biological sciences, we've got many, many different programs. And let me tell you what these programs are. When you did GCSEs, you would have remembered that you covered the processes of life, the 7 processes of life. Some of them are, some schools remember that as Mrs. Gren, the acronym. Those 7 processes of life are essentially what biological sciences is in any degree program. If you're interested in those 7 processes of life, when you think about molecules within cells, then you could be doing a degree in biochemistry or pharmacology. If you think about those processes of life within tissues, then you could be thinking about many, many, many tissues of the body and many, many different organ systems. If you consider that within the humans, within the human body, then biomedical sciences is what you may be interested in. If you want to focus on the brain and how the brain carries out the 7 processes of life and how it coordinates the rest of the systems in the body, then you might be thinking about neurosciences. If you're interested in that from the perspective of plants and animals, then you think about biology, which is exceptionally broad. But if you're interested in all these processes of life at each one of those levels, in animals in particular, then you might be interested in zoology. But if we move to the next slide, why would anybody want to study these 7 processes of life? Let me take you back to what I was thinking when I was an 18-year-old. My aspirations as an 18-year-old were, I wanted to learn about the human body, and I wanted to learn about human disease. And I wanted to do that because I wanted to help humanity by treating disease. And my teachers told me if that's what my aspirations were, I should do medicine. Next slide, please. So I worked very hard. Click again, please, Neil. I worked very hard, and I got into a degree to do medicine at St. George's Medical School in 1991. And had I remained following that path, I would have qualified as a physician 6 years later. However, within 2 months of starting that degree, I realized that there were some things that I would not be able to do when in fulfilling my aspirations as a physician. So instead, I switched over to a degree in biomedical sciences. Neil? I did the degree in biomedical sciences. I started that in 1992, and then I carried on to do a PhD in neurosciences and graduated as a scientist in 1998. So what I want to tell you now is what's the difference between doing a degree in medicine and doing a degree in biomedical sciences if the aspirations are the same. Next slide, please, Neil. So remember what my ambition was. My ambition had been to learn more about human disease and to help humanity by treating disease. And let's take this example here of Alzheimer's disease. The picture on the right-hand side is a picture of Auguste D. She was the first recorded case of someone who had Alzheimer's disease. She was diagnosed or reported to have this disease in about 1906. And on the right-hand side, you've got two images of brains. The bottom one taken from somebody who'd been in their mid-70s who died naturally and didn't really have any disease. And you see the brain is very nice and plump and there's lots of networks in the brain, whereas the brain above is the brain of somebody like Auguste D who would have had Alzheimer's disease. And you see it's really shriveled up, there's lots of cell death, and that's probably why she has those clinical symptoms of Alzheimer's disease. Keep the same disease in mind, and now think about what I would have done as a physician. What would have been my ambition as a physician if I was faced with a person who had Alzheimer's disease like Auguste D? I would have been limited to diagnosing her disease based on clinical symptoms. This is something that I would have learned in my course, and during the clinical rotations, I would have had lots of examples of what Alzheimer's disease looks like, and I'd have used those clinical symptoms to help me diagnose the disease. And then I would treat the disease based on what I've been told works for treating this disease. How is that different to if I was not a physician? And now that I'm a scientist, next slide, please, Neil. What can I do instead? As a scientist, I can still learn about this disease, but as a scientist, I'm now in a position to ask questions about why and how this disease appeared in the first place. I can ask how those nerve cells are dying. I can ask how I can use that information to create novel diagnostics to actually treat the disease. It might shock you to learn that many diseases of the brain, like Alzheimer's disease and Parkinson's disease, actually start in the brain up to 15 years before the clinical symptoms appear. Our brain is so clever that it masks those diseases in our brain, and we don't realize we've got the disease because the clinical symptoms don't emerge until 80% of those circuits in many diseases are already dead. So what we want is diagnostics that can pick up the disease many, many, many years before the clinical symptoms appear. And we also want to be able to use our knowledge of how and why the disease appears to devise novel drugs. I hope you'd agree with me that I would still be fulfilling my ambitions to learn about the disease and to help treat the disease, but I'm doing it in an entirely different way as a scientist. Let me give you a real example of what I did in my trajectory as a scientist. Next slide, please, Neil. So, as a scientist, I asked myself in my lab, during the course of my career, in my lab, I've specialized in Alzheimer's disease, and I've asked myself 4 questions. The first question relates to looking deep in the brain of someone who has Alzheimer's disease and identifying structures that are not found in normal brains. Those structures there, those flame-shaped dark structures there, are called tangles. Those very dark structures don't appear in normal brains. Those tangles are found only in sick neurons in Alzheimer's disease. They're made up of a protein called tau. You don't need to worry about what this protein is, but it's a protein that all of us have in our brains. And the aim here was to ask, if we all have this protein called tau, how does the tau form these rope-like structures called tangles that make nerve cells sick in Alzheimer's disease? That was the first question that a scientist like myself asks. A scientist can then ask, okay, if we want to find out how the abnormal tau forms, how it makes nerve cells sick, the second question is we can ask how it then kills the nerve cells so that you get all of that extensive cell death, that shriveling up of the brain. The third question we can ask is how does that lead to the symptoms of Alzheimer's disease? How does it lead to memory loss? And the fourth question is, how can we use this knowledge to create therapies that can then treat disease? So as a scientist, you want to really delve deep in the disease and ask these questions, but you first need a model. You need a model within which you can ask these questions or test these hypotheses. Next question, please, Neil. And so the hypothesis we had, the hypothesis that we tested in my lab, is that we wanted to answer the first. I'm just going to, because I've got limited time, I'm going to tell you the story of the studies we did to answer that first question. We wanted to ask, how does the abnormal tau, when it becomes abnormal, how does it make nerve cells sick? And we tested a hypothesis which suggested that the first thing that might happen when the normal protein becomes sick is it can't do its normal job properly, and that may be how the nerve cell gets sick. So we asked ourselves, what does tau normally do in nerve cells? Tau is found within the axons of neurons, and you all know about neurons, and you know that the axons are the highways of neurons within which there are tracks, a little bit like railway tracks, along which transport of very important cargo occurs. So things like neurotransmitters that allow the nerve cell to communicate with its target are transported down these tracks. Tau normally keeps these stacks stable. These tracks normally break down all the time. I don't know why nature should devise it like that, but the tracks break down all the time normally. When tau is normal, the tracks are held together very tightly. So we hypothesized that when tau starts to become abnormal in the brains of people who have Alzheimer's disease, the tracks should break down, transport should become disrupted. Next slide. So in our animal model, we recreated flies, we recreated cargo that had green fluorescent proteins within the cargo, within the vesicles that are being transported. We did this in transgenic fruit flies, the brains of fruit flies, because they were easy to manipulate transgenically. And you can see on the left-hand side axons that have come from healthy brains, and the green fluorescent protein is distributed equally all through the axon, which is telling you that you've got very, very nice transport of green fluorescent cargo through that circuit. On the right-hand side, you see what happens if you've got abnormal tau. The red there is just a picture beneath of abnormal tau. It's found all over that sick neuron. And actually, if you were sitting in front of me, I'd ask you, what do you think is happening on the right-hand side compared to the left-hand side? Because you're not sitting in front of me, I'll answer it for you. We think there's lots of traffic jams, pile-ups of those cargo within those sick neurons. We thought that these pileups must occur because the tracks are broken down. Next slide. We used high ultra structural microscopy, high magnification microscopy to look at the tracks. We used electron microscopy. We cut through the nerve bundle so imagine this nerve bundle is coming through the screen, and we cut through a cross section of the nerve bundle, and those little cylindrical structures you see on the left-hand side, those are those tracks that are found in control animals. And you see, if you've got cylindrical structures there, the tracks are intact. This is a track taken, this is a nerve bundle taken from a control animal where there are no traffic jams, and you see those nice intact tracks. If you look at an animal in the next slide, where you've got this huge amounts of traffic jams and pileups, you now see very few of those intact tracks. So our hypothesis was right. We've learned about disease. We've understood that this might be how those nerves are getting sick. The nerves are not dying, they're just getting sick. And they're sick because the first thing that's happening to them is their tracks can't be stabilized because the tau is not abnormal, is not normal. Next slide, please. So how would you use this information to devise a therapy? Next slide, please, Neil. What we decided was, can we sellotape those tracks together? So if the tau is not doing its job, can we bring something else that might sellotape the tracks? We tried to give the animals a drug called paclitaxel to ask if the transport would be recovered. Next slide, please, Neil. And we found that, again, if you look, there are animals where the tau is abnormal on the left-hand side, and on the right-hand side, next slide, we've got slides from animals. These are tracks from animals where they've been fed this drug for only 24 hours. And you see how there's a remarkable improvement in the tracks. And we did many other experiments to show that the transport improved, the behavior of the animals improved. Just one 24-hour treatment with this drug. Next slide, please, Neil. So if we now take this back to my ambitions, I've still managed to, through the course of this study, learn about human disease. I've still been able to do something that may help humanity, but I've done that by learning more about the disease through models that I've created. And I've been giving you a story here, an example of one study, but as I've listed in that trajectory there, over the course of the last 20 years, we've identified multiple ways in which nerve cells get sick, ways in which nerve cells die. That has allowed us to identify drug targets and novel diagnostics to be able to create treatments, which we'll then hand over to the clinicians who will treat people. But you'd agree with me that I'm still solving my aspirations. I'm still being able to meet my aspirations to learn disease and to help humanity by treating disease. And my next couple of slides are just telling you that this is what I did. Next slide, please, Neil. This is what I did, but many other people can use a degree in biomedical sciences, neurosciences, pharmacology, within biological sciences. They've been able to use that knowledge and the skills you gain by going into many, many other fields, which Neil will tell you about in a minute. And you will see that I've given you one story of how knowledge of disease processes and of normal processes can take you one way and through many other different career destinations. But Neil's now going to tell you how, if you studied those processes, but this time in animals, where can that take you? Over to you, Neil.
Okay, hello everybody. It's very nice to meet you. Thank you both so much, Amrit. That was lovely. I've certainly learned something this afternoon. So biological sciences, everything from molecules to cells to tissues to organs to organisms to ecosystems. And this is the area of biological sciences that I work in. I am going to talk to you about the biology and zoology programs. I'm also going to talk to you a little bit about my journey and why I'm doing what I'm doing. I think that, well, let's start here. Okay, so our lecturers, we have a really diverse group of people across our school. And as Amrit has alluded to, we have people who are working on developmental biology, we're working on neurosciences, we're putting all of these things together. But I'm down here in this area, ecology and evolution. And lots of people will tell you that, well, I will tell you that evolution underpins, it's the mechanism that gives us a subject, an area of biological sciences. Before we had evolution, this mechanism for how life is as it is, we didn't have a subject of biology. We had just so stories, the way things got to be, oh, that was just accepted. Ecology is the flip side of the coin of evolution, and ecology and evolution working together have given us our organisms, our ecosystems, and everything that we see in the natural world today. Why am I doing this? Why am I interested in this? Well, if you can believe it, this elderly face here is not as old as you'd think it is. And if you take it back to 1979, 3-year-old Neil was sat down in front of the television by his mum and watched Life on Earth with David Attenborough, who is this gentleman over here on the left-hand side of your screen. And David Attenborough has basically been the voice of biology and science for my entire life. So I was extremely excited to meet him about a year and a half ago, and talk to him about this wonderful skull, which is in the centre of your screen, which is the pliosaur from the Kimmeridge deposit down in Dorset. This is a 2-metre skull of an apex predator, and you can see a lovely representation of it down in the right-hand corner of your screen. This is a huge predatory marine reptile that would have swum through the seas about 158 million years ago, eating everything in its path. David Attenborough was the voice of science for me, and I've been inspired to understand the natural world a little bit better through my entire life. So what have I been working on? I thought I'd give you an overview of some of the stories, some of the projects that I've worked on that I've included undergraduate students in biological sciences in. And I've had students who have been authors on my papers. So first of all, let's talk to you about these two animals here. This is Ceratoseucops. It's a spinosaur. Here's another spinosaur, pretty similar. It's called Ripara veneta. Ceratoseucops is the horned crocodile face, and Ripara veneta is the riverbank hunter. And these are dinosaurs. They're a little bit weird. They've got pointed teeth like crocodiles, We know from another specimen that was found in Surrey in 1983, the first dinosaur I remember being discovered in Britain. It had fish scales, acid-etched fish scales, inside its belly region. And the acid etching is very important. It's not just that a dinosaur had washed into a deposit and then a dead fish had washed in with it as well. The acid etching shows that this dinosaur had been eating those fish and those scales were partially digested. These are really weird dinosaurs. Dinosaurs are terrestrial animals, but these animals are not only eating other dinosaurs. In Baryonyx's stomach as well was found the tail vertebra of a baby iguanodon. They are eating fish, and that's really weird. So we found two on the Isle of Wight, because the Isle of Wight, which is, if you imagine the map of Britain, you know exactly what it looks like. Down at the bottom, there's a lovely diamond-shaped island. It's the Isle of Wight, and on the south coast of the island, it is 125 million years old, thereabouts, and it is the best deposit in the whole of Europe for finding dinosaurs. And we found two spinosaurs there in about 2016. We named them in 2021. Just along the beach, slightly younger deposit, about 120 million years ago old. The water is moving into the Isle of Wight now. The sea is coming in. It's going to switch by 116 million years ago. Europe is just a collection of small islands in a shallow, warm sea. And 120 million years ago, as this water in the ocean is ingressing and we have to have this marine habitat taking over, we have still got a few dinosaurs. And what we found was tail vertebrae, spinal vertebrae, and some other little bits of shin bone and leg bone, which we could identify as being another spinosaur. Another weird fish-eating dinosaur. This animal was about 13 meters long. It is the largest predatory dinosaur that we've ever found from the Isle of Wight. We're working on all of these things all the time. I'm incredibly proud because over the summer, during the lockdown, I had undergraduate students come into the lab, nicely spread out, and we looked at all of these bones, described them, and 3 undergraduate students ended up as authors on this paper. It goes a little bit further, though. What is it about these dinosaurs, these weird fish-eating dinosaurs? Dinosaurs eat other dinosaurs. Dinosaurs eat vegetation. They don't eat fish, except these ones. It's really odd. So we looked at the brain and we were trying to work out what is it that's given these spinosaurs these weird abilities to catch and eat fish. And it turns out, when you look at the brain, they've got a brain that's exactly like any other theropod dinosaur. Their eyesight isn't particularly better. Their balance isn't particularly better. Their sensory structures in the face, this big, thick yellow one here, the yellow nerve canal here, this is the trigeminal nerve. That's the nerve that innervates your face and allows you to feel your fingers touching it. If you've got long whiskers, it allows you to detect if you're pushing your head through too small a hole, like a cat. But this brain is just like a theropod dinosaur brain. And so now we know that the ability to catch fish and process it is not some great neurological innovation. It's a long, thin jaw with pointed circular teeth like a crocodile. And so actually, if you were to give T-Rex a long, thin face with circular teeth like a crocodile, it could take advantage of fish as well. Then we're moving on to another project. This is something that my PhD student Abby is working on. We're looking at bird bone from living birds, because when we go to the fossil record, we want to understand population structure, population dynamics. We can't even tell at the moment the difference between males and females. But the beautiful thing is dinosaurs are the ancestors of birds. So maybe birds can tell us something about dinosaur biology. So we're looking at a special type of bone which female birds produced. It's a sacrificial bone. It's there temporarily. It is used as a calcium source for making eggs. And it appears at the time of producing eggs. So if you've got a bone with medullary bone in it, you know that that is a bone from a female bird. If you have a bird bone that doesn't have medullary bone in it, that doesn't tell you that it's definitely a male, but it does tell you that it's not a female producing eggs at any one time. But it gives you some idea for the first time of what the population structure is in birds. So we're examining that. And again, I have undergraduates and master's students involved in this project. This is my absolute favourite. 1979, little Neil is sitting watching the television and David Attenborough is trekking through the Amazon basin, and there's this weird, stupid looking bird that's just fallen out of its nest, and then it's climbing its way back up with little claws on its fingers. It's called a huatzin. It's a huatzin or a stink bird. They're not very smelly, they're pretty smelly and disgusting, but they live in the trees above the Amazon and Orinoco basins. The parents make rather poorly constructed nests, and surprise, surprise, the chicks fall out. And they fall into waters that are full of piranhas and caiman and all sorts of things. And so to get out and avoid being eaten, they clamber their way back up, and they use the little claws on their wings. And automatically, the link was made in life on Earth to Archaeopteryx. And Archaeopteryx is the first bird. And we've been working on a project. And again, undergraduate, PhD students, all sorts of people have been working on this. And we've demonstrated a model which would allow Archaeopteryx to take off. We've had the ground up and the trees down, so birds running along, or their ancestors running along, flapping their wings, trying to catch prey, and oh, they're flying. Or they're in a tree or up atop of a cliff and they jump off, oh, they're flying. Both of those things don't really work with natural selection. However, if you jump and flap, and small ancestors of birds, the dinosaurs, the theropods, we know were small and covered in feathers. If you jump and flap, you extend your jump. Why would you want to jump? Well, if you're a small theropod dinosaur, you would make a tasty snack for a larger dinosaur. So this all fits with natural selection. If you avoid being eaten and you survive, you've got a good chance of passing on your genes and surviving. If you are able to jump and extend your jump and survive, you pass on that trait. Over time, your feathers become asymmetrical and you add in your flap, and before you know it, you're not just jumping a reasonable distance, you're flying. So that's what we think happened with Archaeopteryx. And it's one of the most exciting things for me, because this is one of the most important fossils that got me interested in doing what I'm doing today. Then we've got the dodo. This is a very exciting project at the moment. We had a paper that was published about 3 months ago, and currently it's still bouncing around in the media. I've never had anything like this before. But the dodo is a weird oceanic pigeon that has settled itself in Mauritius, got big, and then was eaten and destroyed, gone, it's extinct. It's the first animal that we recorded as being present and then recorded as being completely absent and missing. Where'd it gone? And that didn't quite fit with the ideas of the early 18th century, because God had made the world, God had made all the species, and all the species were there, permanent. Only God could get rid of things in like floods. And then we proved it that wasn't the case. We could get rid of animals and plants and all sorts of things as well. But the dodo is depicted as this fat, stupid animal, deserved to go extinct, and it should be eaten. Actually, it turns out it just wasn't prepared to meet humans and cats and rats and pigs and goats. So we are trying to reconstruct the dodo in the most accurate way possible, so that we can think about how it would have fit into its ecosystem in Mauritius, in the hope that maybe we can mitigate some of the ecosystem and habitat loss that's happening in Mauritius now. Last two projects. Mammals. Teeth are really exciting. In mammals, teeth are produced as infants, juveniles, and then they're lost, giving you an adult set. And they are stuck onto a ligament, the periodontal ligament, by something called cementum. And cementum is absolutely crucial. It's laid down in rings every year, so you can age that tooth post-replacement, and you can get this idea. These little animals here, these are from the end Triassic, earliest Jurassic, about 206 million years ago. We've got Cunitherium and Morganeucodon, and you can take their teeth and you can laser scan them, and you can get the marks, little scratches from their last meal, and we can tell that Morganeucodon ate beetles and Cunitherium ate soft-bodied scorpion fly-like animals. But what's really exciting is you can take them and you can count the rings of cementum using high-energy X-rays. And what we have in Morganeucodon, this is a little tiny mammal about the size of a shrew running around in the footsteps of dinosaurs. We have 14 increments of cementum showing that these animals live for 14 years. If you take a shrew today and count its cementum, you might be lucky to get 2. You can also take the long bones and you can have a look at the size of the blood vessels, which are clearly supplying muscles and give you an idea of how active these animals are. And Morganeucodon has blood vessels which are somewhere between modern mammals of its size and lizards. So this is an animal, we can get an idea now of its metabolic rate and its physiological outputs. And this is an animal which is more like its reptilian ancestors than its mammalian descendants, which is all jolly exciting. So all the stuff I've talked about at the moment has been looking at fossils, hard, rocky things. This is what I'm working on as well. This is one of the squidgiest, weirdest structures in the world. This is a placenta, but this is not the placenta of a human. If you imagine a human placenta, it's a disc of tissue, umbilicus, fetus at the end. This is a very weird membrane with these cotyledons. There's an umbilicus here. At the end of that will be a giraffe fetus. We've looked at all kinds of different placentas, and it turns out that they are vastly different across the mammals. They've evolved from a single ancestral mammalian type, but they've evolved into all sorts of different forms, and we're trying to work that out now. And again, we're involving undergraduate students. I have 2 undergraduate students working on this project. I have master's students, and I have a postdoc. It's really, really exciting. So that's what I'm doing. This is just some of the weird stuff. I did a degree in botany and zoology, and this is, biology, and this is where it's taken me. I have an absolutely wonderful time. I enjoy what I do immensely, and I get to work with exciting people all the time. Lots of different careers, lots of different things. There's a whole list here. If you can imagine something that you can do with any degree, you can do it with biology, okay? We're going to teach you all the transferable skills for you to be able to leave Southampton and go into any career you can imagine, okay? Anything at all. However, here are a few of our students who have gone on to do some exciting things specifically so that we can give you an idea. I would also add that within 6 months to a year of graduating, 99% of our graduates are in full-time employment or in full-time further education of higher degrees. So this is Jessica. She's now a program manager for fisheries management. Okay, she's absolutely, she came and did zoology here. And she's got all the skills she needs to now go out and work in fisheries, which are incredibly important, not just for our food and for what have you, but actually these fisheries support ecosystems in the areas where you find them. And these are vitally important parts of marine habitats. Joe Mattock, absolutely wonderful. He's gone off to Padworth College and he's teaching. This is another area that we see a lot of our students going into, going and addressing questions and inspiring the next generation. We've got people who go off, who do the more biomedical side of things, who go off and do medical studies, go off and do postgraduate medicine. And one of our degrees, our biomedical degree, our neuroscience degree, that the biology even, lots of our students go off onto graduate medicine. Then we've got Michael Stefanou. He's gone on and he's also another one of our medics. And I'm really proud of Nat. I taught her in 2013 for a little tiny while, and I have nothing to do with her success. However, Nat is absolutely brilliant. And if any of you have access to Netflix, watch Our Oceans. It starts tonight. It's narrated by a chap, what's his name? Barack Obama. I think he was in American politics for a while. He's narrating this absolutely wonderful natural history documentary on our oceans. Nat was a senior researcher on that project. It's currently it's on billboards all through Times Square in New York. Nat is now an assistant producer making wildlife documentaries at BBC Studios. And these are just some of the people who've gone on from biological sciences in Southampton and have gone on to do really quite amazing things. Here are some contacts for Amrit. And if you've got any other questions, we would be absolutely delighted to take them.
Neil, there are 2 questions there in the audience for you. Sorry, in the chat.
Okay,
I'll read out the question.
I'll stop sharing and we can.
Yeah.
Hello there.
Okay, so there are 3 questions which you could have a go at answering. Would you like to read them out before you answer?
I can't see them. I'm very sorry.
Okay, I'll read them out to you, Neil.
Okay, yeah, that's super. Thank you.
Have you tried analyzing crocodiles' bones? I know they aren't exactly descended from dinosaurs, but they've still lived alongside them. So this must mean that you can learn something about dinosaurs if you look at them, right?
Absolutely. Very good question. So crocodiles here, dinosaurs and birds here. Okay, so they're sister taxa. So in fact, birds here, dinosaurs here. Start again. Birds here, crocodiles here, dinosaurs somewhere in between birds. So yes, absolutely. Although crocodiles aren't dinosaurs, We use them as proxies, and we can look at their teeth and we can see what they're doing, how they're feeding, what they're eating. We can get a good idea as well from their neuroanatomy, and we can relate that to dinosaurs, and we're absolutely doing that at the moment. Excellent question.
Another question. What can I do in year 10 to help me become a zoologist, hopefully?
Okay, year 10. What you need to do is make sure that you're doing maths, that you're doing science. Hopefully triple science would be great for you. You don't have to do triple science. I didn't do triple science. I did double science. But you have a good maths, science background. You need your English. You need to be able to communicate. So all of these are really important. Get your way through. When you come into year 11, pass your GCSEs. This is all easy. It's all very straightforward so far. Pass GCSEs and choose your sciences for A-level, okay? If you want to come and do zoology here, you would need to have a A-level in biology, another science, and a third A-level, okay? But if you've got biology and chemistry, say, and then anything else, you'd be sorted. No problems at all.
Okay, so there are questions coming in, so we'll be quick with our answers. The next question, Neil, for you is, are there any differences in zoology and biology and natural sciences?
Yes. Okay, so biology and zoology, we have a common first year. So we get you all up to speed because you're coming from all sorts of different schools. You will have done different exam boards. You will have had slightly different things coming to get you here. We will give you a common first year. So that gets you right up to speed. So you're then ready to go into your second year. In your second year for biology, you have to do plant sciences, you have to do evolution, and you have to do statistics. Then you've got 5 choices. So eight modules in total. If you do zoology at the moment, you have to do evolution, you have to do statistics, you have to do vertebrate zoology, vertebrate development, animal behavior, and a field course. And then you have 2 choices. Oh, why have I only got 2 choices? You would have chosen those modules anyway. But by giving, as a zoologist, if we didn't make them compulsory, anyone across the university could come in and choose those as optional modules. And then you don't get your seat in those classes. So by making those compulsory, that means that you are guaranteed to be able to come in and do the modules that you want to do. I'm a biologist, I want to do. Yes, there's plenty of seats, you can do those as well. Biology is a nice broad degree. It gives you the option to actually do some of the neuroscience, pharmacology, biochemistry, biomedical sciences options as well. Biology is the broadest of the degrees that we offer. I want to go on a field course. That's okay. You can do the field course as well. So you can, with biology, you can mold your degree more to what you want it to be. You can make it quite general. You can make it quite focused. Zoology, we keep the animal focus, but there's still those 2 optional modules in the second year, which allow you to have some sort of diversity of experience. In the third year, all bets are off. We don't have any compulsory modules, apart from the fact that you have to do 2 modules worth of third-year project, which is a research project. It could be a lab project. It could be an education project. It could be all sorts of different things that we've got going to make sure that you can model your experience to the thing that you want to do. And from day one, you will have an academic personal tutor, or even a personal academic tutor, and your personal academic, I've confused myself. Your personal academic tutor will be there for you for 3 years or 4 years if you do the MSci, and we will help you with your choices, make sure that you're keeping up with things. Anything that you want to do, you come and see us. I have an open door policy, mainly because I don't have a door. But seriously, I have an open door policy. If you need to see me, you can either come up to my office, and if I'm there, that's great.
Neil, I need to get you to answer some other questions.
Yes, certainly. Otherwise, make a phone call, make an appointment, boom, see me.
Okay. Could you tell us about opportunities for travel abroad when doing fieldwork in degrees like biology and like zoology and ecology?
Okay, so
and the next question is, what are the differences between ecology and zoology?
Okay, right. So ecology is a broad degree that allows you to understand the way that organisms interact with each other. Zoology is specifically about the way that animals interact. Now, you might still do some plant sciences because animals are important, plants are important to animals, and understanding how that all works might inform your zoology more. In terms of the opportunities for travel, all of the first years on biology, zoology go to Nettlecombe in Somerset, which doesn't sound very exotic, it's very exotic. And you have a field course down there. But in the second year, all of the zoologists and most of the biologists who want to, all of the biologists who want to actually, go to the south of Spain for a field course, which is included as part of your course fee at the moment. And as far as I know, it's going to stay that way. And that allows you to go and experience a completely different ecosystem. South of Spain is actually desert. Is a desert. It rains less than you'd expect. So you get to see all kinds of different animals, all kinds of different plants of animals, and how they're interacting with one another. In the third year, there is the option to go to Belize on a very exciting field course. So there's lots of travel.
I'm just going to, sorry, I'm just going to interrupt here. I know there's lots of questions coming in. I'm conscious that we're into the final 3 minutes. Lots of the questions are exploring entry requirements, and we'll include the link for students to the University of Southampton's website after this webinar, where they can see the entry requirements for different courses. There was an interesting question that I'd highlighted that came through from a student when they signed up to this AMRIT, and it's related to medical careers that I thought might be a nice one to touch on. And it's asking, what are the real challenges of the journey to get into a dream medicine career, and how can you best approach that process?
Okay, so first and foremost, I'd like to say, George Robson, who's also on the call, if you can put both Neil and my email addresses in the chat, please, so that we can at least, please email us all of these questions that we've not answered you, and we will reply before tomorrow, or first thing tomorrow. You will get responses from us because we're conscious that we've not answered your questions. Right, the question about whether you can do, so in many countries, America is an example, you can't do a medical degree without doing a first degree in biomedical sciences. So if you did a first degree in biomedical sciences, you could have not got into medicine the first time around, or you're not sure if you want to do medicine. You could do a degree in biomedical sciences, and then there are many, many, many opportunities to go on to do medicine. You don't get in after the first year, you get in after you graduate. But in the 25 years that I've been a personal tutor, there is one example of a student who didn't get into medicine, and he didn't get into anything. Every other student that I've tutored has gotten into medicine, and some of them have gotten into medical programs that are highly competitive. The medical program, postgraduate medical program in Oxford only takes 20 students, and one of our students managed to get onto that program. So it prepares you as a scientist, and then you can apply for medicine. I want to just tell you something quickly about our entry requirements. Our entry requirements are 2 A's and a B for our bachelor's program and 3 A's for our integrated master's program. But if you put us down as your first choice, in the 10 years that I've been an admissions tutor, other than the one COVID year when there were teacher assessed grades, we've always been able to take students who missed by 1 or 2 grades. So very often we've been able to take students who've put us down as their first choice if they've got 3 Bs or the equivalent of 3 Bs or the IB equivalent of 3 Bs. We do that before we go into clearing, so that when we go into clearing, we've got a limited number of places, and then we may not be taking those grades. We may be asking for higher grades from the students that are in clearing. There's another question there. What makes biological sciences in Southampton unique compared to other universities? An excellent question. Thank you for asking that question. You should think about whether the university supports you. There are many universities, many Russell Group universities that will offer courses that look very similar to ours. But think about whether you're taught by the people who are actually doing that cutting edge research. I am a neuroscientist. I work on Alzheimer's disease. Neil is a paleontologist. He does the work he's described to you. We are the ones who give the lectures and teach the students and inspire and enthuse students about these courses. You're not taught by people who have to read around that subject to teach you. The scientists of tomorrow, you are the scientists of tomorrow. You should be taught by the scientists of today. That is a critical key thing about Southampton. We don't pawn our teaching to people who are not doing research. All of our scientists are invariably carrying out research. So you're taught by people who are actively doing the things that are important. They know what the questions that are not answered, and they know what should be done to answer those questions, and they pass it over to you. It's a bit like one flame lights other flames. And the second thing that's very important to us, both Neil and I, is our relationship with our students. Your success is our success. I've been to other places for biomedical sciences, you know already from my profile, I never got the pastoral care, the support that we offer. I will never turn away any student of mine who asks me any question, who wants support from me, because I want them to succeed. So you have to ask yourself, will the place I go to give me that kind of pastoral care, ranging from the head of school to the associate dean? I'm the associate dean now in my position. I still have personal duties because I care to contribute to the well-being and the success of our students. There's a lot of questions there that are asking me about what that you guys should do to prepare. Join societies, the British Neuroscience Association, the Biochemical Society, the Royal Society for Biology. These are excellent societies to join. They will tell you a little bit more about these degree programs and where they can take you. And we don't need you to do any work experience to apply to us. What we need to do is hear, why are you interested in this degree? And from reading leaflets from such organizations, you will learn a little bit more about these degree programs to help you populate your personal statements. We will never reject you based on your personal statement. The personal statement is a means for us to learn more about why you want to come to do the course you want to do. We will never reject you based on your personal statement. Please do email Neil and I cannot stress it enough. We will respond personally to your emails. I will do that today before I go to bed, so that you will have your answer, and you don't have to be limited to one question to me. You can email me many times, many times, and Neil will be the same. You can email me throughout the year at different stages of your journey, and we'll always respond.
That is an incredibly kind offer, and one that I know that students will really appreciate, to have that support in what can be a really overwhelming time for decision making and their next steps. We will have to round off today's webinar here. When I email students with the recording, I will include your emails as well. Sorry, Amrit, what were you saying?
Just one final thing. You asked what was unique about us, and this is an example. We are exceptionally accessible, right? So I will pass my email to anybody and everybody, because I care that you make the right decision. Even if you decide to go elsewhere, I'm happy to respond to your emails, and so is Neil.
Fantastic. Thank you both so much. We'll include all of that information in the email we send out to students. I know they'll be incredibly appreciative of all of the information and support that you've offered throughout today's webinar, and hopefully we'll be in touch with you by email, or maybe you'll see them at an open day coming up too, which is a fantastic opportunity for students to find out more about the university, see the campus, which is fantastic, hear about the course, and chat to you both in person too. But we'll have to leave it there for today. A huge thank you to the students that have joined us, and a massive thank you, Neil and Amrit, for your time this afternoon. Hopefully we'll see you all on a Unifrog session again soon.
Thank you very much.
Thank you.